Something's disordered doesn't mean it's dysfunctional. Just ask IDRs or intrinsically disordered regions of proteins. Maybe not so much my hair. Thankfully, there are tools like IUPred prediction plots that allow us to kind of predict what regions of proteins might be in IDR. These regions are important because they can do things like serve as scaffolding, bring different proteins together. They can even promote phase separation, where you kind of have this loosey-goosey protein kind of bring together a bunch of different proteins into this kind of globby thing that's like a membrane-bound compartment inside of a cell, and so it kind of locally concentrates things. And so in order to predict what proteins might be intrinsically disordered, either in big stretches or in little places, scientists can use tools that kind of look at the structure, at the sequence, and predict whether or not it will be disordered. Now, these tools can also be really helpful for people like structural biologists who are trying to figure out which regions of a protein might be IDRs that could be preventing them from getting, say, a crystal structure. And this can allow them to design mutations that'll then remove those regions so they can get a sturdier structure they can look at, things like this. So let's talk more about IDRs and IDR prediction and how to interpret one of those IUPred plots.
You can think about proteins as having these beautiful folded up shapes with things like alpha-helixes and beta-strands that can team up into these beta-pleated sheets, these accordion-like structures. And a lot of proteins are like this, or at least they're like this over most of the protein's length. But there are also proteins that are almost entirely disordered. And even those really nice structured proteins might have regions of disorder, say in between those helices and those strands. By disorder, I mean that it doesn't have a set structure. It's more like loosey-goosey. And intrinsically disordered, basically, this is telling us that the sequence of the protein can provide the information that we need in order to know that it's going to be disordered. So on its own, it might be disordered, but it might actually adopt some structure when it binds to something. That's one of the things that these IDRs, or intrinsically disordered regions, can do. And that's not all of them do that, though. However, there's a bunch of different functions for these proteins and these regions of proteins. They can serve as scaffolds and bring multiple things together. Sometimes what you have is in addition to having IDRs, or intrinsically disordered regions, which might be interspersed with more strong structures, you can actually have IDPs, or intrinsically disordered proteins, which have these really large strands, if not entirely disordered regions. And these can do things like promote liquid-liquid phase separation, where basically they bring together various proteins, various molecules, maybe DNA and RNA, all these sorts of things into this kind of gloopy glob that locally concentrates things to promote reactions and that sort of thing. As the name intrinsically disordered suggests, this means that basically the sequence of that region or of that protein is enough to tell us that in the absence of anything else around it, it's likely to have no set structure. It's likely to be disordered. And this means that we're going to be able to predict which regions of a protein might be disordered and what proteins might be IDPs, or those intrinsically disordered proteins. So I'll tell you a little bit about the theory, and then we'll look at some examples of how you can use this software to generate the IUPred scores and plots and all this stuff, and for any protein that you're interested in. So first, though, I want to tell you about what these scores actually are. So our goal is to predict unstructured regions of a protein. And how do you do this? Well, there's some information based on the known disorder regions of proteins. If we're talking about experimentally validated disordered regions of proteins, there's this database called DisProt, which is a collection of these manually curated. So basically people have said, I used this experiment and we found that this region was disordered. And so then that information goes into this database. And so you can find this curated information about disordered proteins here. And if you go and you look into the disordered proteins or the disordered regions of proteins from the experimental data, you'll notice some things. And one of the things that people noticed early on is that there tend to be depleted of nonpolar and hydrophobic amino acids and rich and charged in polar amino acids, so hydrophilic things. But this isn't nearly enough information. Instead, we need more information. And so there's this software called IUPred. And what it does, so that stands for like intrinsically unstructured scores prediction. And basically, instead of just going off of that, okay, well, these amino acids are more likely found in unstructured regions of proteins, what they do is they actually look at structured proteins. So they look at proteins that are going to be like in the PDB, so Sol structures of proteins where they have crystal structures, they have cryo-EM structures, this sort of thing. And they know that a region of a protein is well structured. And then they look and they see, okay, well, what does that structure, are the elements of the structured regions, and how does our sequence compare to those? So what IUPred does is it takes the amino acid sequence of a protein, and then it's going to predict what regions of the protein are going to be intrinsically disordered or not have a defined structure. And how it's actually going to do this is kind of all mathematically technical, but it uses this thing called an energy estimation algorithm. And it's basically, from the best I can tell, tries to figure out which amino acids are happy hanging out together in a structured protein. I'm using this like energy estimation matrix. So they go through those known structured regions of proteins, and then they look and see what amino acids are near one another and calculate like the free energy, or how comfy those amino acids are next to each other, how happy they are next to each other. And then they take the sequence of the protein of interest and they say, okay, in this protein, we don't have the structure, but things that the amino acids that are next to it and near it in the sequence are probably going to be near it in the structure. So how likely are we to find those together in a structured region? How happy would they be together in a structured region? And if it kind of matches with the profile of a structured region, then we're going to say that it's likely to be structured. And if it doesn't, it's likely to be unstructured. And so if you want to look at all the math and stuff, it's there in the paper, but I am not a mathy type of person, so I will leave that to you and just go by having you know that the IUPred score is going to be a probability. It's going to be a probability at the amino acid level. So you're going to see a plot that's going to be plotted over the length of the protein when you're looking at an IUPred plot. For each individual amino acid, you get a score, whether it ranges from 0, which is likely structured, to 1, which is likely disordered. And this is basically, remember, it's going to be calculated based on that amino acid identity. So what is that actual amino acid? And then what is the surrounding protein context? What amino acids are near it in the sequence? Again, we don't have a structure, so we can't say, okay, what would likely be near it in the structural form, but we know that the things that are near it in the sequence are likely to be near it in the structure. And so then we just need to know, like, are they happy hanging out together, or are they going to want to be disordered if they're near each other? And then we can predict whether they're likely structured, in which case we'll find it below the line, or disordered, in which case we'll find it above the line. Similarly to how when I was talking about the hydropathy plots, we're going to do some sort of smoothing. So instead of just looking at the amino acid, we're going to look a little bit around it when we're plotting these out so we can kind of smooth things. And we'll see how we can smooth things at different levels, depending on how noisy we want our data to be. If we have a protein that's going to be really, really well structured, most of it's going to be below that line. You might see a few parts above the line, and we'll look at this a little later. These parts are often located at the ends, but most of it is going to be well structured. It's going to be well ordered, and you're going to see it below that line. What about an intrinsically disordered protein, one of those IDPs? This is going to be mostly, if not entirely, above the line, and we'll look at some examples of that. But a lot of the time, what you have is you have a lot of proteins that have large or medium-sized regions of disorder, and these are going to be predicted just have predicted disorder, so things above the line, but they're also going to have most of it's going to be below the line. And let's look at some examples of this as well. So as I mentioned, we're going to be using this IUPred 3 software. So this is there's been multiple iterations of IUPred, and the latest version, from what I can tell, it offers a couple different things like more smoothing options. They also have some features I'm not really going to talk about. There's this thing called Anchor, which is going to predict context-dependent disordered regions. So disordered binding regions, which basically are going to be regions that are going to do things like change shape when they bind to a partner. So they undergo kind of like a conformational change where they go from being unstructured to having a set structure. And this anchor tool is going to be able to kind of predict that sort of thing. They also have a tool that can kind of see like whether it might have a structure in an oxidized versus a reducing environment and things like this, as well as some conservation. So you can check out those things if you want to know more, but I'm not really going to talk about that too much. What I do want to talk about, though, is some distinct examples so you know what to look like, what it looks like, and then we'll go and we'll take a field trip to the IUPred site, and I'll show you how you can actually do it for any protein. But first, some examples. So this is an example of one of those IDPs. And you can see that most of it is above the line. This is telling us that it's likely to be disordered. Again, this is a probability. So when you look at one of these things, it's not telling you it actually is disordered, and it's not kind of like an extent of how ordered or how disordered it is. It's a probability. So some things either basically it has a set structure or it doesn't. And some you'll see it above the line, it's more likely. Below the line, it's less likely. But we're looking at kind of like a yes, no, is it disordered or does it have a set structure? And the higher it is above the axis, the more likely it is to be disordered. And so this is really, really likely to be disordered over a lot of its length. And what are we looking at here? We're looking at this protein called TNRC6A, which I commonly refer to as GW182 because it's part of this GW182 family. And basically, it's involved in something that I studied in my grad school, which is called RNA interference. And I go way over it in a bunch of other posts. But basically, there's this protein argonaut, and it takes these little short RNA pieces, about 20 nucleotides long. This can be a microRNA or an siRNA. And it uses it as a guide to go and find messenger RNAs with a complementary sequence. It then binds to that sequence and causes it to be degraded and stop protein from being made from it. So it's a way that cells can kind of regulate protein production, and it's an important method that's used in the lab to experimentally like knock down genes. But when our bodies use it, we don't really use siRNA naturally, but we do use microRNA. And so microRNA, basically, when it goes and it binds to that messenger RNA, it needs help in order to get it degraded. And how it gets this help is it binds to the scaffolding protein GW182. And that's the protein that we were just looking at, which is this largely disordered protein. And by having all this disorder, it's basically able to bind to a lot of different things. It has binding sites for all sorts of different things. And it's really flexible, so it can kind of get those things where they need to be. And it can hold on to decapping complexes and de-adenylation complexes. So kind of be in contact with both ends of a messenger RNA and help degrade it. And so really, really cool stuff with GW182. And it's one of those IDPs. So that was an example of a protein that is basically disordered over most of its length. But important proteins can have large IDRs, but not themselves be IDPs. An example of this is P53. P53 is one of those proteins that's really, really highly mutated in a lot of different cancers. It basically can do all sorts of things, including sense cell damage and kind of stop the cell from dividing over and over before things get out of control. And so P53 has large disordered regions. As I mentioned, there are different levels of smoothing that you can do. So what we're looking at there was medium level. But you can also see if you have no smoothing, it's going to be really, really jaggedy. If you have high smoothing, then you're going to have a lot smoother of a line, but you might miss some of the local features if you're like averaging things out too much. But I'm going to use the medium smoothing, which is the default in the next few slides to point out a few different things. One is that those IDRs are located in the structure at the N and the C termini, so the beginning and the end of the protein. And this is really common. IDRs are often located near termini. So with P53, we had these disordered regions that were really, really large. But even well-structured, mostly well-structured proteins can have short disordered regions. And an example of this is going to be that Argonaut protein that I was telling you about. And so this Argonaut protein, you can see that it's mostly has this strong structure. But what you can see is you can also, if you see regions where there's these dotted lines, and they actually right here, there's a whole missing segment. Basically, these regions are going to be local regions of disorder that you don't see in the structure. This is a crystal structure solved by crystallography, and you actually don't see those regions in the structure because they're too disordered. What the structural biologists are able to do is they're able to kind of take a sort of snapshot and look at the structure of a protein. But if a region of the protein doesn't have structure, it kind of just averages out. And so you're not going to see that in the structure. And so even though it's there in the protein, we can't see it in the structure. And I'll go into this a little more later. But you can see that it largely does have these structures, but then you have these regions that are unstructured. And if we look at the IUPred plot of this protein of Argonaut, what we're going to see is that we see this mostly structured, but we also see some regions that are predicted to be non-structured or intrinsically disordered. And these regions are kind of better seen if you use a short, the short modeling method. Basically, IUPred has a couple of different settings, and so you can do it with a long setting or a short setting. With the long setting, it's kind of looking for larger regions of protein disorder, whereas with the shorter region, it's looking for smaller regions of shorter regions of protein disorder, such as those things that you might see missing in a protein prediction in a protein structure. And so you can see here that at the termini, now we have predicted disorder at both of these ends, whereas with the long structural prediction, we didn't really see it as much. And in fact, if we look at the structure of Argonaut, as we'll do later, we'll actually see that we can't see the this N terminus in the structure because it's going to be disordered. Another thing I wanted to point out was that below the actual plot, you're going to see a few different things in the IUPred server. And these are kind of like different annotations from different databases that also show you things about what's going on at different places in the structure. One of these things that you can see is PFAM. And PFAM is going to tell you about conserved domains. So protein domains are kind of like rooms in an apartment. And so they can be like distinct structural domains. So like here you can see that there's these, I've colored it by the domains. You have these different regions of the proteins, which kind of have these defined structures where they're kind of often separated by a little bit of a more flexible region. And these are going to be protein domains. And so we can have functional domains too, which basically are regions of the proteins that do different things. And so really domain is just a name that we kind of give to help us refer to parts of a protein. And these domains are often going to be well conserved between proteins of between similar proteins in different species, as well as multiple versions of that protein in a cell. And so much more about this thing in other posts, but basically we can classify these domains and we can characterize these domains and we can look at all these different conservation of domains and versions of the protein and how the domains are similar or different and all this types of stuff with domains. And then it goes into this database. And so there's this PFAM database, and then that information gets plotted alongside your IUPred prediction plot or below the IUPred prediction plot. And what you can see is that these conserved domains are often but not always structured. And so those unstructured regions aren't in those conserved places in a lot of things. Another thing I wanted to point out is that post-translational modification sites, so things like phosphorylation sites, are often located in IDRs. And sometimes having the modification actually changes the structure. But having that like disorderedness makes it kind of, you can think of it kind of making it easier for it to get modified. lots of flexibility for those enzymes to come in to modify it, all sorts of cool stuff. But anyway, so in this protein Argonaut, what we're looking at here is there's going to be this predicted disordered region with a bunch of these phosphorylation sites. So when we're looking at this PTM, that stands for post-translational modification sites. And here there's this cluster of phosphorylation sites, and this is actually what I studied in grad school. So I was studying how this region gets phosphorylated by a protein called casein kinase 1 alpha, CK1 alpha. And this phosphorylation actually affects the activity of Argonaut. It causes it to kind of release that messenger RNA after it's done working with it, we think, regulating the protein's activity. And if you want to know more about it, you can check out my eLife paper. It's in eLife, so it's open access. Anybody can read it and look at it. And I also have posts on it and things like that. So hope that you find it interesting. And I drew it as a dotted line here because it's actually missing in that structure. So we couldn't see it in that protein, which goes back to what I was talking about before, was that how the IDRs are actually often unresolved and thus unmodeled in protein structures. Because remember, when we're solving a structure, which is with a method like cryo-EM or X-ray crystallography, what we're looking at is, well, structure. And so we need the proteins, like in the case of crystallography, we need the proteins all to be stuck in the same conformation, the same shape. And those regions of the proteins that are intrinsically disordered, they're not going to want to be stuck in a single state. They're going to be wanting to be moving all around. And they're going to kind of average each other out. Or even worse, what they can do is they can prevent us from getting a structure of a protein because they're so loosey-goosey. They're kind of moving around and making the rest of the protein not want to stay still either. And so sometimes what crystallographers, we want to know what sites or what regions are likely to be not unpredicted, not only so we can not expect to see them in our structures, but also so that maybe we can actually chop off those regions to have a better chance of getting a protein to crystallize. So you don't typically want to just chop off in the middle of a protein, but because those IDRs are often located at the termini, we can often like truncate a protein, so cut off the ends to make it more likely to crystallize. Even in the case of a technique such as cryo-EM, where we don't actually have to get the protein to crystallize, we just have to have it, we just freeze it and then like look at them all in place. If you have an intrinsically disordered region, all the different copies of the protein in each copy, the region's likely to be in a different side of shape, a different conformation. And so then they're kind of going to average each other out, and you're not going to see them in the structure either. So bottom line, don't expect to see IDRs in a structure because these regions of the protein are going to be different in each of the different copies of the protein. And so you have to have a lot of them in the same orientation in order to be able to see them. So much more about how we experimentally solve structures in other posts, but this structural information, or the lack of it, can also be used as evidence for disorder. So if we have a region that's missing in a crystal structure, this can actually be used as this experimental disorder proof. And so if you look at that DisProt database, you can see that for Argonaut protein, it'll actually show that the region of the protein, this region of the protein is disordered, and our evidence is that it's missing in all of these crystal structures. So it was present in the protein sequence, that it was actually physically present, but we can't see it in the structure. And therefore, this is evidence that it's disordered. So now I'll take a quick field trip and look at how we can actually use these tools. So IUPred's website looks something like this. It's going to give you the option to put in a protein identifier, which we can get from like IUPred, or to just paste an amino acid sequence in there. And so we'll start by looking at that P53 protein I mentioned. So basically, if I just Google search P53 IUPred, I can find the protein. And now I can just copy and paste this accession number into the IUPred. And now basically it gives me this option where I can choose whether I want what level of smoothing I want. Do I want to have it predicting for long disordered or for short disordered? Anchor, remember that's the binding regions that are predicted to be disordered binding regions. So they bind something maybe and then they take on a shape. And so various options. They also have that redox state, conservation, so you can stick in aligned sequences and things like this. All sorts of different things I'm not going to go into, but these are where you would find those different options. I'm just going to go with the default medium smoothing. And if I submit this, now it'll take a second, but what we're going to see is it's going to give us that plot, and it's going to have those annotations below it that is going to provide information about the protein. So here we can see those above the line, we have the regions that are predicted to be disordered. Below the line, regions that are predicted to be ordered. We have experimental evidence that these regions are disordered. These regions also have a bunch of post-translational modifications. You can see that this large structured region is going to be one of those domains in PFAB, PPAM, and things like this. You can also basically, if you go to show structures, you might be like, oh, wow, it has structures over the entire length of the protein. But what you're looking at in a lot of these cases is when you're looking at something in one of these disordered regions, what you're looking at is going to actually be like an NMR structure. So if we go to this 2YL4, you can see that it's an NMR structure, which basically is a solution method that works good for small flexible things. And what you get is this ensemble. So because you have the protein in all these different orientations, you kind of get a look at different snapshots of those orientations as this protein is moving all around. And so you can see that those regions of the protein might be solved with NMR. And then you have regions that are going to be, but the majority of the structures you're going to see are of that region in this middle that has that structured domain. And again, if we go and we were to change the smoothing, we can go from no smoothing, medium, high. So those are a few of the things that we can see in the structure of P53. There's also this really nice paper I'll link to where basically they talk about how they used a bunch of different methods in order to try to figure out information about the different regions, including those non-structured regions. And what's pretty cool is basically they say like more than 30% of eukaryotic genes encode contiguous unfolded regions longer than 30 amino acids in length. And so basically a lot of our proteins are going to be have large regions of intrinsic disorder, so have those IDRs. So what about my favorite protein, AGO2? So I do the same thing, but I take the accession code for AGO2, and now I get this prediction. And you can see that this is the region I was talking about that I studied, where you have this cluster of phosphorylation sites, and it's located in this region that is both predicted to be disordered and shown to have evidence of disorder. And so if you're wondering, okay, well, what's the evidence for this? Remember that DisProt database? If I were to go and I were to take the same accession code that I put into IUPred 3, and if I put it into DisProt, well, now I should have the protein pop up. If I click on it, it's going to tell us more information. And so you can see that only 5% of it's going to be disordered. And if you look at basically the evidence for disorder, you can see that this region is missing coordinate residues in in the structures. So in all these structures, it's missing these regions. You also see that the N-terminus is also missing. Remember, that was another region that was predicted to be disordered, especially when we use the short method. And we see evidence for this disorder in the absence of it in the structure as well. So basically, there are a lot of different things that you can see using DisProt as well. What about if we look at the actual structure? So there are a couple of ways that we can get there. So if we just put that accession code into UniProt, which might be where we got the accession code, if you go down to structure, it'll link you to PDBs, so the protein database with solved structures of it. It'll also, so some of the times it's not the default one might be like a structure of just a single domain. The one we were looking at before was going to be for F3T, which was the structure solved in Limor Joshua Torres' lab by Elado Kayam, who I worked with. And this is the structure solved to of Argonaut 2, human Argonaut 2 bound to a microRNA. And so if we look at the structure in the PDB, we go to the 3D view, what we'll see is that when you see residues, so these amino acids that are going to be like missing, they're going to be grayed out. They're missing in the structure, but they're present in the sequence. And they'll also show us these dotted lines. Sometimes you might have a really long disordered region that's going to get cut off. And so like it's above the cutoff. And so if it's above the cut, basically if something's above the cutoff, it's long enough that a long enough missing region, it might not actually show a dotted line. And so again, so what I'm doing is I'm just viewing it with the integrated viewer. But if you really want a good look, you're going to want to go into something like PyMol or something like Chimera X to better manipulate these things. But it's easier to just view it here if you're not familiar with those tools. But again, you're going to see that so that like 8, this region here, the region I was interested in, is going to be missing in the structure. And so it shows up as this dotted line. And it's grayed out in the sequence. There are also sometimes when you have something that's actually going to be missing in the sequence. So they took that part out of the protein, such as when we're talking about one of those truncations. And so if you go to the structure, then that would show up as well, the mutations. But here, basically what it's telling us is that these regions are just unmodeled. So we can see that these regions are unmodeled here. And you have a disorder prediction here as well, telling us that that's likely to be disordered. And indeed, that fits with what we were looking at before. A word of caution about if you're using AlphaFold, it's going to try to predict a structure for all parts of the protein, including any IDRs. And so when you see an IDR in an AlphaFold structure, it's often going to be represented as kind of just a line. You might look kind of like spaghetti. And often it's going to be colored like dark orange, which means that it's very low confidence. And so that would be another indicator of an IDR, is that the software can't predict the structure for it. But again, this isn't true experimental evidence. For more information about how IUPred works, I found this article really helpful, this prediction of protein disorder based on IUPred. So this is the 2018 version. So basically, it's it's the original IUPred. And so now we're on IUPred 3, and IUPred 3 has that paper I showed you before, and I'll link to both of them. But this one really goes over nicely the kind of what's going on behind the scenes of the software. IUPred is just one of the methods for just predicting disordered regions of proteins. There are a lot of other ones. IUPred is one of the main ones. But if you just Google search, Wikipedia will show you a bunch of other ones. And what you'll see is that they're based on different methods. They might be better for different things. And so here's a list of some of them. Here, this is a 2021 open access. So again, anybody can look at it. A Nature Methods paper that kind of compares different methods and talks a little bit about what's good for different things, because the prediction tool might vary. What's better for predicting one type of disordered region might not be the same, might not be the best for another type of disordered region.